Clathrin is a protein that facilitates endocytosis. We're going to Google, what is endocytosis? Endocytosis is an active transport process where eukaryotic cells engulf external substances, such as nutrients, pathogens, or fluid, by wrapping them in the portion of the plasma membrane, which then pinches off to form an internal vesicle. Now let's go over here. Clathrin smuggles quantum dots into living cells. Peptide coatings can help nanoparticles slip into cells, a process that may prove useful for in vivo imaging and drug delivery if scientists can clear up how it works. Now, Abdulaziz, Mitsuru, and co-workers with Japan's National Institute of Advanced Industrial Science and Technology have a better idea after thoroughly studying the insect neuropeptide allostatin and learning it helps with calcium selenide/zinc sulfide quantum dots pass through cell membranes by recruiting clathrin, a protein that facilitates endocytosis by forming vesicles around foreign substances. The researchers suspected that allostatin might be gaining access to cells via galanin receptors. But when they blocked those receptors, it didn't have much influence on the influx of quantum dots. By inhibiting PI3K, a kinase that is crucial to the formation of clathrin vesicles, they noted that fewer than half of the quantum dots made their way in, leading the team to conclude that most of the nanoparticle entry is clathrin-mediated. The remainder of the quantum dots may be entering through electrostatic interactions with molecules in the cell membrane, thanks to an arginine residue on allostatin, they suggest. So, clathrin smuggles quantum dots into living cells in 2009. A neuropeptide helps slip quantum dots through cell membranes by recruiting clathrin. Has any nanotechnology been updated since 2009? Because it's 2026. It's 17 years later. Now let's go back to Google, and we're going to search. Is clathrin the only protein that facilitates endocytosis? No, clathrin is not the only protein that facilitates endocytosis. While clathrin-mediated endocytosis is the best characterized pathway, cells utilize multiple mechanisms for taking up materials, many of which do not require clathrin. These are collectively known as clathrin-independent endocytosis pathways. Clathrin-independent endocytosis. That's about the different DNA, different environments, different types of beam steering, and molecular communication in the atmosphere for sensor networks, for all sorts of reasons, which occurs without the involvement of clathrin. Now let's scroll down and click on this link. Clathrin-independent pathways of endocytosis. There are many pathways of endocytosis at the cell surface that apparently operate at the same time. With the advent of new molecular genetic and imaging tools, an understanding of the different ways by which a cell may endocytose cargo is increasing by leaps and bounds. In this review, we explore pathways of endocytosis that occur in the absence of clathrin. These are referred to as clathrin-independent endocytosis. Here we primarily focus on those pathways that function at the small scale, in which some have distinct coats and others function in the absence of specific coated intermediates. We follow the trafficking itineraries of the material endocytosed by these pathways and finally discuss the functional roles that these pathways play in cell and tissue physiology. It is likely that these pathways will play key roles in the regulation of plasma membrane area and tension, and also control the availability of membrane during cell migration. So, in the routing for molecular communication, the little molecule, you turn it off with a binary, you turn it on with a binary, and you watch it with the quantum, and that's all done through the wireless sensor networks. Basic one source data. You don't need a quantum dot in your tissue to see you, view you, or change you.